Australia. Dr. Brewer is a plant biologist who specializes in molecular biology, cell biology, and plant physiology. His recent research has focused on production and action of plant hormone, in particular the complex interaction between strigolactone and auxin. Strigolactone are a regulator of shoot branching, root architecture, and plant fungi symbiosis. They are promoted by auxin and help crop plant cope with suboptimal growth conditions such as low nutrient, shedding, cold, drought, and salinity, and attack from pathogen or parasitic weeds. Strigolactone also inhibit auxin transport. A key goal of Professor Brewer is to uncover the mechanism of plant hormone function and to understand how this mechanism influence plant architecture and environmental response in crops. So its seminar title today is Lateral Branching Oxidoreductase Act in a strigolactone biosynthesis, some pathway that specifically inhibit filtering in barley. So thank you, and we are excited to listen to you.
Fantastic. Thanks very much for that introduction, and thanks for the invite to give a talk. I'm really pleased to present some of the latest work that we've been publishing on the strigolactone pathway in barley. And but first I'd like to give a little bit of background on, just going to get to the next slide, on the background on how we're working on phytohormones. So our aim is to try to employ or manipulate the phytohormone pathways in order to provide some sort of a trade outcome for crops. And when we're working in this topic, we have to consider that not one hormone just doesn't do one job, and it always acts together with a group of different hormones. So in this case, as an example, we have intact pea plant with a tiny bud in the axil of the leaf here, and it's in a repressed state because it's repressed by a combination of hormonal signals all acting together, almost like a little equation here, in order to get a trait outcome or a phenotypic outcome at that node. So auxin travelling down from the main shoot tip provides information to this node that there is a growing shoot tip above, and that this bud should be not growing out. And the way that it does that is it works through the promotion of strigolactone, which we've been part of the discovery of strigolactone as the branching inhibitor hormone. The strigolactone then moves into that bud and blocks it from growing out by activating things like the transcription factor TB1, or T-center branched 1, which is a cell cycle inhibitor. At the same time, though, auxin represses cytokinin, and cytokinins are a class of growth promoting hormone, and that by limiting their production in this node region, then that bud gets a double message to say don't grow out. But in some other sort of context, say for instance when the shoot tip has been removed, then the equation is a different equation. You've got you're missing the signal of auxin. You no longer have strigolactone production, and you have a de-repressed cytokinin production. So that means that that this bud will grow out, and in fact the whole shoot tip kind of replaces the one that was missing. So when you're studying a particular hormone pathway like strigolactones, we always need to think about the context in the other signals involved, and this is simplified. There are quite a few other signals involved in this decision making. One of the other problems with working with phytohormones is that they have a range of different phenotypes, and they act together with the other hormones on multiple different phenotypes. So for example, strigolactone is the branching inhibitor, as I mentioned, But strigolactones also, they promote senescence, they regulate stem thickness, they have a role in plant height, and they modulate or modify the root system architecture, and they are exuded into the soil to promote fungal symbiosis. So you can imagine that if you want to utilize or modify strigolactone pathway in order to change the branching habit, perhaps to get more or less, more yield or some other effect, then you're going to consider the effects on all of the other traits. And we do see that in the strigolactone mutants, where they're dwarf and bushy and stay green, and there are root architecture changes. So even though you might have double the branches and double the seeds or grains, you do end up having potentially a yield drag based on the other phenotypes that are affected. Alternatively, you might think of increasing the strigolactone pathway, which would repress branching more. But, and in this case, this does happen naturally when you have a situation of low nutrients, then the branching is repressed by strigolactone. And, but then you have to consider that you'll get more senescence, changes in the root architecture, and more in fungal symbiosis potentially. So these can also be a bit of a negative drag on yield. So how do we solve all of these problems? That's part of the research that we've been doing, or perhaps the basis of the research we've been doing in barley, and coming at it from other species like Arabidopsis. So today I just want to focus in on one part of the strigolactone pathway in order to kind of show you potentially how we can do this. So, the strigolactone pathway involves biosynthesis. It starts with beta-carotene in the plastid. A few enzymes, MAX3, MAX4, are involved in producing this common precursor called carlactone. It's not bioactive, but it is mobile. So it moves out of the plastid, and it can move out of the cell and upwards in the plant to carry the strigolactone signal. When it arrives at its destination, it is then converted by other enzymes into a hormone or a hormone precursor. In this case, it's methylgalactanoate that we think this is the main hormone precursor that's common in all plants. But this carlactone can also be converted, particularly we think in the root system, to other strigolactones. And in rice, as an example, the carlactone is converted into orobancol, which is then exuded from the root system. And this is the classical four-ringed strigolactone, or canonical four-ringed strigolactone shape, whereas methylgalactanoate has these two rings. It's a little bit different. So one thing that we notice in all the different species of plants and crops that have been investigated is that each plant and crop species produces a different type of strigolactone mix. They can be these canonical strigolactones with the four rings, or they can be kind of some more unusual shapes. And most of these are in the root exudate of that particular species. So this does open up some avenues where can we kind of mine these biosynthesis pathways to get a more trait-specific outcome and avoid those pleiotropic issues that we get with a stronger type of mutant that completely eliminates strigolactones. So what we've done in the past is done some reverse genetics, and this enables us to find other components of the strigolactone pathway So in this case, we did a big experiment in Arabidopsis at the University of Queensland when I was a postdoc with Christine Beveridge. And this was a great experiment because we could find other components of the genes in the pathway. And we used mutants, in strigolactone mutants, which have increased feedback on their own expression and production of strigolactone. So those pathway genes go up. In a decapitation situation and other treatments, we get another effect on the strigolactone gene pathway. So that allowed us to detect other genes which followed the same pattern, not necessarily the amplitude, but that pattern up and down here, which was enabled us to find this new gene, which we called ELBO at the time. It's a 2-oxoglutarate dependent dioxygenase. There's a lot of these in Arabidopsis that are doing all sorts of different jobs as an enzyme pathways. And in this case, ELBO is represented as mostly as a single copy in all seed plants. So the next thing in Arabidopsis is reverse genetics is to get the mutant, you order it from the stock center, and it turns up and you grow it up and you look at the phenotype. And in this case, we had a slightly dwarfer, bushier mutant, not as strong as the kind of the stronger strigolactone mutant UMAX4. So it's an intermediate mutant, which probably wouldn't have been found in a forward genetic screen. So there is value for reverse genetics. And it's not additive with the other pathway in the biosynthesis pathway mutants. And the expression pattern is the GUS expression is in the vasculature all through the plant. And this is where we see the other biosynthesis genes being expressed and producing strigolactone all through the plant in the vasculature, down into the root tip and up into the buds. So the work I won't go into in detail today, but just to summarize, is working with Kaori Yonayama in Japan on the chemistry and biochemistry of the pathway, a range of different experiments we did, and including expressing the ELBO gene in E. coli and feeding it with different precursor compounds, also labeled precursor compounds, and measuring the products that are produced. And we discovered that ELBO makes this variant on methylgalactanoate, hydroxyl methylgalactanoate. So we think that this is either the branching hormone or another precursor to another branching hormone downstream. It's been very difficult to work on this because it breaks down very quickly and in solution, and it's very difficult yet enough to properly work with this compound or analyze it. Work is continuing to discover what the branching hormone, the branching type of strigolactone is in Japan at the moment. So just switching then gear, coming a bit forward into the future, I really wanted to work on barley as a model crop to try to explore if we can initially modify tillering to change and improve yield and other effects of crop production. So I'm at the time moved to Adelaide University at the Waite campus and worked with Artie Sitamaran to create gene-edited mutants in barley of various parts of the strigolactone pathway. I'll just talk today about ELBO. So Artie and we got some nice gene-edited mutants, and we also learnt from Laura and Marek that an ELBO allele, which has got a mutation in the active catalytic domain, also Laura was working on that. So we joined forces, came together and put together this publication, which was published recently on the ELBO mutant and its effects in barley. So just like in Arabidopsis, this mutant is intermediate from the stronger ones that we see in barley that Marek has discovered. We'll talk a little bit more about that. So we think that this is a kind of a sub-pathway in barley that produces the strigolactone branching hormone, but doesn't have as many of these side effects that the stronger mutant previously isolated, the receptor mutant called D14. That's got a higher rate of tillering and a range of other defects which make it tricky to work with in terms of crop development. But elbow has perhaps more potential. The tillering is more, but it's not as extreme as D14 and has other things which are normal about it. In fact, when we tried to look at with Carrie, work with Carrie and myself, when we tried to look at the strigolactone production in the root system of these mutant alleles, we found that the roots actually produce normal levels of strigolactones. In this case, we've got barley lactone too. Barley makes these unusual types of strigolactones, which were just discovered in recent times. And elbow is making them, which is quite a surprise. And when we looked at mycorrhizal colonization, this is the symbiosis with the fungal, with the fungus. That was also normal in these mutant alleles. Very strange that the plant is not failing to make a branching strigolactone, but it's making other strigolactones as normal. So we wanted to explore this in a bit more detail, and we know that under some stress conditions, strigolactone levels go up, and this causes a branching repression. And we see this quite clearly in barley. Barley is quite plastic. It will respond well to different stress, or badly to different stress, I guess, depends on your point of view. And in this case, say some sort of suboptimal condition like low nutrients, the tillering is greatly reduced, and ultimately this affects grain number and yield. So we're thinking that, well, what would happen if if the strigolactone pathway wasn't repressing tillering so much? Can we get a level of yield stability, or less plasticity? And how would that translate to yield? So we wanted to just knock out the tillering effects and see what would happen. So barley elbow is really good for this. In this case, this is just an example of what we've been working on, and perhaps you heard Jack Kelly's talk recently on this mutant D14, which is the receptor mutant, and it's dwarf and bushy and stay green, just like a strigolactone mutant in all other species. And pushing it into a low nitrate condition, we do see the plant reacting, but not as strongly as the wild type. So there is a partial stability or lack of plasticity in the mutant, which ultimately could preserve yield in a stress condition. So we did the same sort of experiment with elbow mutants. And we're wondering, the question is, are we hitting a sweet spot where we can increase yield stability but have fewer of the side effects? And indeed, we are seeing that. This is a glasshouse pot trial with different nitrate rates, and looking at tillers and other trait-related, yield-related trait. And we see that, just have a look at the dark bar for the minute, and you'll see that the wild-type plants respond quite strongly to the reduced nitrate, almost halving the tiller number. And ultimately, this has an impact on grain number, a big impact, and ultimately a big impact on yield per plant. And but having a look at the mutant doesn't respond as strongly. It's resistant to that effect on the nitrate, and we get more tillers, many more tillers under the low nitrate, many more grains per plant, an increased yield of nearly, or not as big a drop of yield. And one of the trade-off, though, that we've discovered is that elbow mutant consistently has smaller average grain size. So this is the big trade-off at the moment that we're thinking about. But there's a huge amount of yield potential there that if we could, find out what's going on with the kernel size, then we could potentially bring both together in lower input type of agriculture. Just having a look at some other traits in the mutant, and you'll notice here that harvest index drops off. So the plant deliberately is investing less into the grains, and the strigolactone is doing that because the harvest index remains high in the mutant. Interesting too, fertility is affected. There's a drop in fertility, and so again the plants are deliberately killing off some of its progeny, and strigolactone having that effect there retains the fertility. So there's a few positive effects. We don't see a lot of change in shoot biomass or the root biomass, so they seem to be pretty consistent, whereas D14, the stronger mutant, will have effects. And the plant height is just slightly smaller. So yeah, I think we're really getting close to a sort of sweet spot for this type of yield stability. So a little checklist here. We've got we've discovered through this elbow pathway, a special strigolactone that is the hormone that is specific for tillers, but specific for tillers and not other traits so much. There are more tillers, more grain. This leads to more stability in yield and in fertility in suboptimal conditions. There's near normal biomass and height, and normal root exudate and symbiosis. So again, we need to have these things normal to not have any kind of yield drag. And but the only deficiency at the moment that we know of is the smaller grain size. And we don't know a lot. We've still got a lot more work to do. We don't know, for example, if there's a drought stress response problem, as has been seen in that receptor mutant D14. So more validation and research is needed, and particularly in the field. So yeah, still in the early days, I guess, for this work. So just to touch for the last few minutes on what's happening with the grain size, and this is work from Jack, who looked at the expression of all of the pathway genes. Maybe he presented that. And this might remind you that the strigolactone signaling genes are all expressed right throughout inflorescence and grain development, the stages. But the biosynthesis pathway is largely absent, except one MAX1 gene and elbow are present, particularly elbow as the inflorescence of grain develops. So we do think that this might indicate that strigolactone precursor carlactone is being delivered to the developing inflorescence, and then elbow is converting it to the hormone on site, where it has an effect on grain development or grain filling. And right at the moment, we're just looking at a bit more of the expression pattern. So coming to La Trobe University, my new home, I've connected with Marta and Matt Lucy, and they've been doing spatial transcriptomics of barley and have a really wonderful resource. And we do see the strigolactone receptor and elbow expressed in specific locations in the developing grain. So this is definitely something happening in there, but it's super confusing because when Jack applied strigolactone directly to developing inflorescence, this is GR24 synthetic strigolactone, he found the opposite from what we expected, that grain mass was repressed. And he also applied a strigolactone biosynthesis inhibitor and got the opposite effect. And he's also replicated this. So it's a real effect with the strigolactone pathway, but it's opposite from what we expected. So we're still working through what that might mean. We've got various theories about it, and Jack's going to finish up his PhD. Hopefully, we'll solve the whole problem. And so there, with that, I'll finish up and just again give my acknowledgement and thanks to some of the key members that have done a lot of this work, and there are others. Sorry, I haven't got everybody here, but I just want to say it's been a fantastic, optimal team to try to optimize barley in the trait of tillering. So I'll finish up there and perhaps take some questions. Thanks.
Thank you, Phil, for your talk. It was very interesting, very comprehensive, and such a nice nice phenotype you are working on, and the potential of it to mitigate some low input and things like this. So now we're going to pass to the question session. People can either choose to ask their question themselves by raising their hand, or to put their question in the chat, and then I will read the question for Phil. So far, I don't see any hand that had been raised, but I see some question in the in the chat box. So if you're ready, we're going to start. The first question comes from Sarah McKim. I feel great talk. Can you clarify if tillers or the main stem or both show smaller grain size? And relatedly, whether there is a difference in grain size between first maturing spike or later maturing spike?
Okay, so I think it's so if you get more, if I can perhaps interpret it's a little bit. Thanks, Sarah. It's great to hear from you. So maybe if I could just interpret that a little bit, and correct me if I'm wrong. So this figure here shows the kind of spread of effects of the grain size. So now with the X-ray CT scanner, we can get really high detailed information of every single grain. So we do see, so this allows us to perhaps separate out pools of different sizes and have a bit more of an understanding of where the small grains and the large grains are, even within each spike. I'm just trying to think of the answer though while I'm talking. So in this case, the GR24, I mean, it did kind of repress, make some pretty small grains down here, but the bulk of them are still on average smaller, and the opposite was true with the inhibitor. So I don't have that data specifically here for elbow, so I can't yet answer that question, but it's a great one because, yeah, we will need to know if there is sort of late forming tillers, perhaps they don't fully mature so well, and then you get that kind of drag of small grains in those compared to the early ones. So that would have to be something that we'll need to answer in the future. Thanks.
Thank you. I have a kind of related question about that overall you observe a smaller grain, but more grain and higher yield so far compared to the wild type. Do you know if it impacts the malt quality for malting in this industry? And how do you feel it can be received by the industry, for example, for feed or malting to have smaller grain but still higher yield? Would it be interesting for the industry?
Yeah, we haven't yet got any data on the malting quality or other quality traits. I think perhaps I think Jack is going to try and do some of that work. We do have the intention to look into this because we really got to keep that malting quality, and this is why we're focusing, I guess, on a little bit at the moment on the problem of elbow with this average small grain, and whether there's going to be some other industry, unwanted effects for industry. At the very least, though, in terms of grain for other uses for barley, it would be, it'd be a thumbs up. But we still don't know about malting quality.
Okay, thank you. So here there is a second question in the chat box. This is from Robbie. I feel great to hear from you. I see you're well. The effect of LBO appear relatively minor, which may not be identified in example mutant screen, knowing the gene. Did you mine natural diversity for variation in LBO in barley? That's the first question. And maybe you can address this one, and then I will ask the second question on his message.
Yeah, let me sort that one out. The short answer is no, not yet, but that would be fantastic to do. It is highly conserved across different species, so we may be it may be a pretty highly sort of fixed type of gene, particularly in its regulation. So it's hard to know how to predict that, but yeah, look, it also looks like it's a rate limiting step for strigolactone acting on tiller number. So in that case, yeah, it could be some variation, which would be very interesting. So if you can help me do that, Robbie, that would be fantastic. What was the second question?
Yes, the second question is, has a tillering phenotype been identified at LBO or D14 in GWAS experiment? Have you molecularly screened, characterized tillering, dwarfing, grain size mutant for variation in either gene or phenotypically under different nutrient conditions?
Again, a really nice question, which I can't answer, not yet. But yeah, it would be fantastic to mine more of the GWAS data in Australia or in any population that we've got. And it's a fantastic idea to potentially run a population under something like a nutrient poor condition or reduced fertilizer regime, and then to see how the response is. So not only do you have like this steady state type of strigolactone effects on tiller number, kind of in optimal conditions, but then you've got the reaction of the strigolactone pathway to the particular stress condition, which these two things could be different. So it would be fantastic to do both types of experiments and look at the GWAS and potentially get quite different genes coming up which are somehow connected or linked with the strigolactone pathway.
It could be very interesting. So if it's help, Robbie was adding in the chat, I can't mute and edit myself, but we could help for this. So if you're interested, you can contact here. May have people that may have interest in collaborating with you.
Robbie, if you wanted to, you could unmute now.
I'm allowed to speak. Phil, lovely to see you, lovely to hear from you. All I was saying was we've got lots of opportunities, I think, to look at this in some of the germplasm that we've characterised over time. So yeah, just drop us an email and we'll see what we can do. Maybe Laura, actually, Laura could also probably help. Or even I see Matt and the Oz Barley team might also be able to give some advice.
Yeah, thanks, Robbie. It'd be great. We can, yeah, we'll definitely get together somehow with various people and try to work towards that.
Oh, I didn't even know Laura was on the call, so I'm glad I mentioned you. Laura wants to speak.
There's Laura.
Hi guys, nice to hear from you. Thank you. Yeah, we were looking at natural allelic variation in this gene years ago, but at the time there was only exon capture, and I think the gene was not covered. But I'm looking now in ensemble plants. I see there's a few variants, but I'm sure, Robbie, you have a wider repository of data that could be mined. And in terms of GWAS, I think the best work I am aware of, at least in terms of genetic diversity, is the work that was done by Gary Muehlbauer a few years ago in Minnesota under field conditions. And he had lots and lots of peaks. So one possibility would be to look if any of those peaks correspond to the position of the gene, and maybe chat with Gary about any potential collocations. I can send you the paper, Phil. That's talking about tailoring, of course. But for sure, as Robbie says, the gene might collocate with QTLs or GWAS peaks for other traits. I'm thinking about the tailoring side of things. Yeah.
Sounds great. Yes.
There's a host of barley grain size mutants, so small round grain, which would be quite interesting to look at, just PCR sequence to see whether or not the genes mutate in any of them.
Yeah, that'd be great. Thanks.
Too many things to do, Phil.
Yeah, I know. Yeah, I'm a little bit distracted on tomato at the moment, unfortunately. I mean, fortunately, yes, I've got at least something to work on.
Okay, there is no additional question. One additional comment that Kelly Houston was adding in the chat, if that may interest you, there is some GWAS data set that she had been involved in this production under a reduced N and nitrogen and have a Taylor number too, so maybe it could be helpful.
Great, thanks, Kelly.
If there is no additional question, I think we're just going to thank you for your great talk and your interesting story you share with us today. I guess it's become late on your side, so we will say you thank you and have a good rest of the day on your side.
Thanks a lot. It's been a pleasure.
Great to see you, Phil.
Thank you, Phil.
Lovely to see you, everyone. Bye for now.
Take care.
Bye. Thank you.
Bye-bye.
Bye.
Thanks, Phil. Well, we'll be in touch, and we'll send the link to the recording if you wanted to use it for yourself at any point.
Great. Thanks, Malcolm.
Okay, take care. Chat soon.
Lovely to see you.
Thank you for your time, Phil. I really appreciate your contribution to this seminar. Bye-bye.
Bye for now.